Hello. For my lecture journal 1, I decided to write about things that I had learned from each chapter that I felt were important. And I also want to tell you my favorite part whenever we were doing lecture was when you referred to Edward Jenner as Caitlyn Jenner. But from chapter 1, I learned that the levels of classification, which I didn't even know was really a thing, honestly, which is probably kind of bad. But the levels of classification being domain, kingdom, phylum, class, order, family, genus, and species. And something that really kind of blew my mind was that viruses are not even actually cells, which probably should have known that too, but how they're microbes and they infect the cells of their host in order to reproduce, which I really did not know that. In chapter 3, I learned the 6 I's for the laboratory techniques, which is probably really important for the future. The inoculation, incubation, isolation, inspection, information gathering, and identification. And then chapter 4, I learned that the non-pathogenic bacterial group, majority of bacterial species are free living and not involved in disease. And then chapter 7, the environmental factors that have the greatest impact on microbes are nutrients, temp, pH, amount of available water, atmospheric gases, light, pressure, and other organisms. And then chapter 8, I had not even heard of fermentation ever, but it is based in the glycolysis pathway in which both electron donor and final electron acceptors are organic compounds. And chapter 9, I couldn't really read on my own because I'm using my sister's old book and she kind of took chapter 9 out, so I'll have to find that. And then chapter 11, I learned the differences between ways of controlling microorganisms, which is decontamination, sterilization, disinfection, and sanitation. And the differences in those because there's a difference. I know that now.